Welcome. In the last session, we studied about muscle fiber or myocyte in detail, and we understood that the muscle fiber inside it are parallelly arranged myofibrils. Now, in this session, we will see the structure of that myofibril. So we know that it is this myofibril that shows alternate dark and light bands. So it shows alternate dark and light bands. So here let's see it how that dark and light bands are seen. So here this is the myofibril. So it will be cylindrical in shape, and you will be seeing, so this is the dark bands. So the dark bands will be present with the white bands. Okay, so this is how we will be seeing this, the dark band, and this is light band. Now let's see this how these dark and light bands are formed. So here when we see inside the myofibril, we find two type of proteins called actin filaments, and one more called myosin filaments. So these actin filaments are thin, whereas the myosin filaments will be thick. So hence they are called thin and thick filaments. So now let us see how that thick and thin filaments. So this arrangement of thick and thin filaments will produce dark and light bands. So you'll see here. Okay, so these are the actin filaments. Okay, so in the last session I have also shown you that how that the actin and myosin filaments are present in a three-dimensional picture. So these are all the actin filaments. So here also you see, and again, so like this you will see continuous the actin filaments and even myosin filaments. Now in between these actin filaments we see the myosin filaments. So these myosin filaments will be thick. So we'll see later why and what makes them to be thick, and why the actin filaments are thin, and what makes them to be thin. So you can see here this is the myosin filament. So here also you will see the myosin filaments. It will continue through the myofibril. So here I am not showing the the whole entire one. We'll just see here one more. So this goes on. Okay, so this is another myosin filament. Again, myosin filament. So here, and on this we find some special structures which will form cross. Okay, so later we'll see how it cross bridges and how it's in the contraction. Okay, So this here I mentioned, this is the actin filament, and this is the myosin filament. So one is actin filament and another one myosin filament. Okay, and here when we see this, okay, here the region, so here from this region, so I'm just mentioning here, okay, so this zigzag line to another line here, okay, will form a functional unit, what we call it a right, uh, the sarcomere, which we'll see here. So here this point, so here where the actin and myosin filaments overlap, so the point where the actin and myosin filaments overlap is what we call the A band. And you can see here one more, okay, so where you find only actin filaments, so there are no myosin filaments. So here you will find again actin filaments. So here also I'm drawing, right, and again here we see, so this one where you find only the actin filaments will form the I band. So it is this I band that appears light in color due to presence of only actin filaments. So this is a light band, whereas the A band where you find myosin and actin filaments will be dark. So this will be the dark band. So you can see again the light band, the dark band. Again, if you come over here, this is also again I band. So this I band is the light band. Again, okay, from this point to this point. So this will be again the A band, right? So here the I, which stands for isotropic, whereas the A stands for anisotropic. So you can see the light band alternating with the dark band, again the light band and the dark band. So this is the light band, this is the dark band, and it continuously goes on. So this is what, okay, so wherever this dark region that we see is the location where the actin filaments and myosin filaments are overlapped, whereas this light band is a region where you find only actin filaments. So now here, okay, in between this where you in the dark band region, in the dark band region where you find only myosin filaments, so from, okay, this point, okay, to this point, so from this point to, okay, this point, so here I'm just mentioning, okay, so this is what we call the H zone. So this is the H zone. So you find the H zone in the A band, so it's the H zone. Now at the center of this A band, so exactly at the center, okay, we find one membrane. So there is a membrane exactly at the center of this A band. So this membrane is called M line. and this is the M line, is M line. So here the M line is thin fibrous membrane, is thin fibrous membrane, right? And now here at the center of I band, at the center of I band, you have is another line. It's called the Z line. So this is a elastic fiber, say elastic fiber, right? So now from this Z line, so you can see again one more Z line, and here also you will see, okay, here again one more Z line. Okay, so from one Z line to another Z line is, so from one Z line to another Z line is what we call a sarcomere, is called sarcomere. So here this is the functional unit of contraction. The sarcomere is functional unit of contraction. So it's a functional unit of contraction, right? So from one Z line to another Z line. So just I'm okay mentioning here. So this is from one Z line to another Z line. Okay, so we have seen in between the I band, that is the light band, we have, okay, Z line, right, which is an elastic fiber, and in the A band we have one thin fibrous membrane, we call it M line. Now in the A band you will see some portion where there is a, okay, overlapping of myosin with the actin, and some portion there is no overlapping of actin with myosin. So here is this region where you see there is no overlapping of actin on the myosin, only you find myosin, and this region is what we call the H zone. Okay, the H zone. Fine. Of course the H zone will get decreased when there is a contraction taking place. So, right, and if very one important point during contraction, so during contraction, right, the length of the sarcomere, length of sarcomere, it decreases. So the length of the sarcomere decreases. So here the sarcomere is from one Z line to, right, another Z line. So this is what I'll mention. So from this point to this point, so this is the length of the sarcomere. It decreases when there is a contraction. Now the length of the A band, so when coming to length of A band, the length of the A band remains same. So the length of the A band remains same. It does not, okay, decrease or increase, whereas the length of the sarcomere decreases, and even the length of the H band also decreases. So here again I'll mention on this side, length of H zone also decreases. So these points are important. The length of H zone decreases, the length of sarcomere decreases, whereas the length of the A band, it remains same. Right. Here I'll even tell you there are other names for this Z line and M line. We'll see some names. Okay, so other names for Z line. So here Z line is also called Krause's membrane, also called Krause's membrane, or also called the Dobbe's line, also called Dobbe's line. So this is a Z line or Krause's membrane or Dobbe's line. Then the H band or H zone. So H zone or band, you can also call this as a Hansen's disk, also called Hansen's disk, right? So this is about the structure of myofibril. Okay, so here from the Z line to Z line is the functional unit of contraction, is what we call it a sarcomere. And we have seen in the sarcomere, when there is a contraction, the length of the sarcomere, that is from Z line to Z line, okay, its length decreases, whereas even the H zone, that is present, okay, in the dark band where there is no overlapping of actin filaments, so there the H zone will also get decreased, and whereas the A band, the length of the A band remains same. So this point's important. So now let us see, okay, the structure of those contractile proteins. So here the actin and myosin are the contractile proteins. So let us see the structure of contractile proteins. So those are actin and myosin. So actin and myosin filaments. Right. So first we'll see about the actin filament. Right. So here we have is actin filament. Now let's see what this actin filament is made up of. This whole actin filament is made up of two F actins. So you can see here this whole one. So this is one, okay, F actin. So this is another F actin. So you find two F actins that helically coil. So the actin filament consists of two F actins helically coiled around each other, coiled around each other, right? So in this case the F stands for filamentous, it stands for filamentous. So two F actins helically coiled here, and each F actin, so this, as I said, that this whole is a F actin. So here not the brown color thread like structure I am talking about, the green color, okay, one that you see, okay, attached one to one, and they are seen helically coiled. So here there are two such F actins, the filamentous actins that are coiled around each other. Now each of these F actin is composed of right, monomeric units called G actins. So each F actin is composed of okay, several monomer, okay, or monomeric G units. So monomeric G units. So you can see this is the G unit. Okay, so this is another G unit, and you can see many G units, they linked one to one to form a long, okay, F actin. So that is a filamentous actin molecule, right? Now running close with these two F actins is another protein that is called tropomyosin. So this is another protein. So this is what we call tropomyosin. So this tropomyosin is also two filaments. So it is two filaments running, okay, along with the right F actin. So there are two F actins, as I mentioned here, two F actins, and also two filaments of tropomyosin. So this is one tropomyosin, this is another tropomyosin, and they are also in close contact with the F actin. Now attached to this tropomyosin, we find, okay, three proteins. You can see here there are three uh, proteins. That's a complex protein, what we call it tropomyosin. So there is a tropomyosin protein attached to the tropomyosin. So this is the tropomyosin, and this tropomyosin is seen attached to the troponin, not to the F actins. So tropomyosin is attached to, okay, I'm sorry, troponin. So troponin, so here I mentioned tropomyosin, I'm sorry, right, troponin. So troponin is attached to, okay, tropomyosin, is attached to tropomyosin. So this is what troponin. Now this troponin is of right, it is made up of three units. So here each troponin is made up of three units. So those three units, or what we call them, okay, TNT, that is troponin T, then TNI, and then we have one more is called TNC. So these are the three units of troponin. So now let's see where these are present. So here the one, in this case, out of these three, so one will be, right, TNT. So this is TNT. Then another one is TNI, and one more is TNC. Okay, we will see here the functions of each of these. So what is the function of the TNT? So the function of the TNT is it binds the other troponin components. It binds the other troponin components to tropomyosin. So it binds the other troponin components to tropomyosin. That is the function of TNT. And coming to TNI, so the function of TNI, it inhibits the myosin. It inhibits the myosin binding site. Myosin binding site on the actin filament. Okay, so the T and I, so as the I stands for, okay, inhibiting the myosin binding site on the actin. So that is right function of T and I, whereas T and T, well, it stands for, okay, binding the troponin components to the tropomyosin. And coming to the last one, the function of TNC. The function of TNC is that it contains binding sites for calcium ions. It has binding sites for calcium ions that will actually initiate the muscle contraction. So that we'll see, okay, how this muscle contraction takes place. Okay, and this troponin and tropomyosin, they are often called regulatory proteins. So they play a very important role. Okay, so I'll just mention here both the troponin and the tropomyosin. So these two are called regulatory proteins. They are called regulatory proteins. Why they are called regulatory proteins? Because they play an important role in masking and unmasking the active sites of the actin. So that is about the structure of okay, actin filament. Now let us see the structure of myosin filament. So the next one about myosin filament. Okay, so here this myosin filament is a polymerized protein. It is a polymerized protein, and each of these myosin filament is made up of many monomeric proteins. So each myosin has many monomeric proteins. So these monomeric proteins are what we call them the meromyosins. So the monomeric proteins are the meromyosins. Okay, and this meromyosin will be having two components. So the meromyosin has two components. One we call it HMM and another one called LMM. The HMM stands for heavy meromyosin. So it's heavy meromyosin, and the LMM stands for the light meromyosin. So let's see its diagram. So this is the myosin filament, right? And this myosin filament consists of two polypeptide chains. So you can see here, okay, two polypeptide chains, and they are wrapped around each other, right? So these you know what you say, the those monomeric proteins, many which together form, okay, meromyosins here. Now when we see each meromyosin, so this is a meromyosin and it has, okay, two important parts. So this is it's a globular head. It's the globular head. Okay, so from this point to this point, so we can say that it's the head, and below that there is a short arm. So below that globular head there is a short arm. Now the rest of this is what we call the tail. So this whole portion we call it tail. Now this head and short arm, the head and short arm together, they form the heavy myromyosin, so that is HMM. So this HMM is the one that actually forms cross arm during muscle contraction, that we will see it, okay, when we see the muscle contraction. So this head and the short arm together constitute heavy myromyosin, okay, and the rest of it is the tail, and this tail is the one that constitutes LMM, so that is light myromyosin. Now the head has two important points, okay. One, there is a site for binding with actin, so this is the site, okay, which binds with the actin filament. Then coming to the other points, so these are the locations where ATP binds. So this is the site for binding of ATP molecule. So because the muscle contraction is a energy requiring process, and hence the energy is obtained from this ATP molecule, that is adenosine triphosphate, okay. So here this globular head, so where as I said, it has a site for binding of ATP. It has an active ATPase enzyme, okay, and has these binding sites for ATP, right. So the globular head is an active ATPase enzyme, okay, right. So and here this when you see here, this is the myromyosin actually. So here from this end to, so wherever I'm drawing the line, so this is one myromyosin, so this is one myromyosin, and I'll draw another one with the color. So this is another myromyosin. So you can see here there are two myromyosins, and many, okay, such, okay, myromyosins together, they will constitute one thick filament. So that's the point here. So many of these many myromyosins together constitutes one thick filament, together constitutes one thick filament. So you can see here two myromyosins are present. So one I have drawn, okay, with black color, and one with an orange color, you can see here, okay, from this point. So this is one myromyosin, and the other one is another myromyosin. So in the earlier diagram we have seen, okay, the actin and myosin filaments together. So you may find so many coiled like this, and that forms the thick filament, that is the myosin filament. So all this will be the tail regions, and from these tail regions you will see the head projecting onto the top. So you will find, okay, so many heads projecting on the top, you can see here, okay. This is how you will be seeing. So this is all, many myromyosins will be held like this, and okay, so all these portions is the tail portions, and what you see on the top is the head portions. So as I mentioned here, so many myosins together constitutes, okay, one thick filament, constitutes one thick filament. Okay, so here in this diagram you are seeing, okay, two myosins which will constitute a myosin filament. So here in this is a head, what you see here, and the remaining whatever you see is the tail. So this head, and there is a one short arm. So this will actually, these two will form the cross arms. The head and the short arm together will form cross arm, which helps in attaching with the right actin filament. Okay, so this is the structure of myosin filament. And before I go here, again I'll show you the actin filament. So this is the actin filament, right? So which later it may bind. So this head will bind with this actin filament. So just mention here actin filament, and of course it is present, these actin filaments will be present all around this, okay, myosin filament. So the next part is, we'll see about triad system. And before I go to discuss the triad system, so here are a few more points. So the myosin filament, so it's a total length, the myosin filaments total length, it will be uniform and is almost exactly, and it is around 1 point so around 1.6 micrometer. So it's 1.6 micrometer. Okay, now what if we take a transverse section through AI band? So how it looks? So the question here, how many actin molecules surround the thick filament, that is a myosin filament? So we'll see that one also. And one more here, an important point, the length of sarcomere. So that's the total, means that the length of the sarcomere is around 2 micrometer. So the length of the sarcomere is 2 micrometer. Okay, and the length of this myosin filament is around 1.6 micrometer. Now we will see the transverse section if we take it through, okay, AI band, and how it looks like. So right here I'm showing you, okay, the transverse section through AI zone. So that is A band and I band zone. So when we take transverse section, how it looks like. So here you can see this thick one is the myosin filament. So this is the thick filament, and it is myosin filament, whereas this green color one is thin filament, and this is called, okay, the actin filament. It's called actin filament. Okay, so now here each thick filament is surrounded by 6 thin filaments, surrounded by 6 thin filaments. So you can see here, okay, 1, okay, 2, the 3, 4 and 5, 6. So you see, okay, so there are 6, okay, thin filaments. So each thick filament is surrounded by 6 thin filaments, okay, whereas each thin filament So each thin filament will have, okay, 3 thick and, okay, 3 thin filaments as neighbors. Okay, so this is about how we take when we take the transverse section through the AI zone. So how this, okay, so this is where I'm showing you in a three-dimensional. Usually whenever we draw it on the board, we see only on one side of it. So we need to see the whole transverse section, then we'll come to know, okay, how it looks like. Okay, so that's the transfer section through AI zone. Now let us see the triad system. So let's see here the triad system. So here the plasma membrane of the muscle fiber, so which we call it sarcolemma. Okay, so this is the sarcolemma, right? And it forms an invagination into the sarcoplasm. Invagination into the sarcoplasm, and this is what we call it a T tubule. So here the T stands for transverse tubule. Now this one on either side, it has uh, the endoplasmic reticulum, and we know the muscle cells have a special type of endoplasmic reticulum, and it is called sarcoplasmic reticulum. So you will find, okay, so the sarcoplasmic reticulum will be seen, so on either side of this T tubule. So here you can see the sarcoplasmic reticulum. Okay, right. And this sarcoplasmic reticulum is a storehouse of calcium ions. So it is a storehouse of calcium ions, and these calcium ions play a very important role in the contraction of muscles, that we will see when we study about sliding filament hypothesis. Okay, right. So T tubule and okay, plus 2 terminal cisternae. So these terminal cisternae, which are part of this endoplasmic reticulum. So these two, the T tubule and the 2 terminal cisternae, together form, okay, the triad system. So what is its function? We will see in the coming session. So how this triad system helps in transmission of a nerve impulse into the muscle fiber, and thereby allowing the release of these calcium ions from this sarcoplasmic reticulum, which will bind with one of the component of the troponin, and that will actually help in contraction. So how it is, we will see in the coming session.